Today, Joanne Yew, an associate professor at the UH, will be presenting her research on what Hawaiian Drosophila, I didn't practice that word before I started, Drosophila can teach us about our microbiome. Her research is being done through a permit with the HETF. Please note that permits are required when entering the HETF for research purposes. Joanne, whenever you're ready, you can start.
Screen. Okay. Hi, everyone. Thanks so much for joining, and I'm really happy to be able to talk about some of the work that we do and how the HETF ties into that and why it's been a really valuable resource for us. So my name's Joanne, and I work on Hawaiian Drosophila, and specifically about their role in ecology and what they can tell us about evolution, and maybe not expectedly how we can learn about our microbiome based on the biology of Hawaiian Drosophila. Hawaiian Drosophila are a remarkable subgenus. You can see here several examples of them, and they have very different kinds of wing patterns and body pigmentation. And this just indicates how diverse they are phenotypically. And that's a large part of the reason why we are interested in them, this kind of rich diversity that is quite rare in other kinds of taxa. So I work at UH, and this is just a shot of UH Manoa, specifically in PBRC, Pacific Biosciences Research Center. So just a few words about what we do. We work on a variety of different kinds of organisms, including corals, Hawaiian bobtail squid, copepods, and mosquitoes, disease ecology of mosquitoes. So these are very diverse organism systems, but one thing they all have in common is that they have a microbiome. So the microbiome is the collection of microbes and their products that reside in or on an organism. And we've known about their existence and their importance to our ecology and health since the 19th century, but it's only been the last 15 years or so, due to several technological innovations, that we've been able to see just exactly how important and pervasive their contributions are to all kinds of organisms and the environment. Within humans, this map here shows a map of different microbial communities that are found in specific body regions throughout the body. And you can see that based on the pie charts here, each of the colors represents a different genus of bacteria, and different colors means there are different bacteria. And you can see how the colors and the pie charts here vary a lot, just from the hand to the knee to the armpit to the feet. So it's been said that the microbiome community in my hand is more similar to that of your hand than it is between my own hand and, say, my own knee. Since making these kinds of maps, we've also discovered that the microbiome has a lot of importance in both our health and disease. So diseases like Crohn's disease, diabetes, certain kinds of cardiovascular diseases, obesity, all these appear to have some sort of microbiome dysbiosis or imbalance to them. And as well with health, as we know, it's quite easy to find probiotic supplements in the grocery store these days. And a good balance of certain types of microbes is thought to aid us in our digestion and even our general well-being, our general psychology and behavior. Other organisms, of course, use the microbiome as well for their physiology. So a good example of this are plants, and specifically plants and microbes, specific microbiome communities in both their roots and their leaves and the flowers. In the roots, in what's called the rhizosphere, the roots here interact with microbes that are found in the soil, and this is both bacteria and fungi. So the image below is just scanning electron micrographs of different kinds of microbes. You can see them intimately connected with the roots of the plants, and they help to support growth, and they provide nutrition for the plant. In other kinds of animals, including insects, microbes found in the gut and in other organs help with digestion, and specifically for the bees. You can see here in this blow up of the gut in fluorescence here are bacteria that help to digest certain kinds of sugars, and this allows the bees to draw nutrients from these sources. So microbiome is pervasive. It benefits, and sometimes it can hurt. Perhaps one of the most striking examples to me of just how important the microbiome function is through the experimental use of germ-free animals. So this is one common way of studying the function of the microbiome, is to basically take an experimental model like a mouse or a fruit fly, and then wipe it clean of its microbes. So this is called making axenic animals or germ-free animals. So now that we have this blank slate, we can implant into the animal different compositions of microbiomes and see what the effect is of particular taxa on the health of the animal. This example from mice shows that if you implant microbes from an obese twin into a germ-free mouse, you end up recapitulating that obese phenotype. Whereas if you do the control where you implant microbes from the non-obese twin into the mouse, you see a normal weight mouse. Similarly, in Drosophila, if you place into Drosophila a mutant strain of bacteria that is unable to metabolize a particular cofactor, in this case, acetyl-CoA, you end up getting a much smaller fly, which has been compromised in its development and growth. So both of these experiments show the direct connection and the sufficiency of the microbiome in contributing to the overall host health and physiology. So what was sort of mind-blowing for me was that microbes can also have a microbiome. So bacterial-fungal interactions and how they interact with each other, that is also a key part of understanding how a microbial community contributes to host health. So certainly in disease, you've probably heard of Candida and Pseudomonas. These two this interkingdom interaction actually increases the virulence of each other. And so this is commonly seen in cystic fibrosis tissue. This sort of interaction has also been implicated in bowel disease and in making burn wounds much slower to heal. In plant health and development, bacterial-fungal synergism also increases plant productivity and nutrient acquisition. And in our day-to-day lives, we encounter the benefits of bacterial-fungal interactions, such as with sourdough starters and the rind of cheese. So it's thought that these interactions contribute a lot to either the rise of the sourdough and also the flavor of bread and cheese. So now that I've kind of given you an overview of all the different types of ways in which the microbiome contributes to our life and to the life of the environment around us, just going to introduce some of the two big questions in the field. One is, how does the microbiome assemble? How does this disparate community of bacteria and fungi and protists and viruses and other kinds of things, how does that assemble inside an organism? What are the influences to that? And second is the functional view. How does the microbiome contribute to host physiology? And people are studying this in all kinds of organisms and in the environment as well. So one big challenge in this field is the microbial complexity. We have hundreds to possibly thousands of different microbes in us. And so it's difficult to tell who is important and who is a tourist, who's just passing through and really doesn't contribute much. And then with so many hundreds of different taxa, how do you pick who to start doing experiments on, or how do you pick which ones you want to use for probiotics? And the second challenge in the field is that with this complexity, then how do you begin to do any sort of experimental manipulation? As I indicated, right now the large amount of data we have is on these snapshots, these pictures of which microbes are where. But how do we move from correlation to actual causation? How do we understand the underlying mechanisms? And that's quite difficult to do with these complex systems. If we are to move into a place where we can start using microbes to treat disease or as a prophylactic, then we have to be able to understand what are the underlying mechanisms. So this is where, at the very basic level, Hawaiian Drosophila are a fantastic model to start to understand, to start to address these questions. So why Hawaiian Drosophila? Hawaiian Drosophila have been an important model of evolution and adaptation since people started studying them, their ecology in the 1950s. And one reason why they're so interesting is because of their explosive adaptive radiation across the Hawaiian Islands. So this image here just shows that from a single species, founder species on the island of Kauai, you have a number of founder events indicated by these arrows to each of the younger and younger islands. And these Drosophila have colonized hundreds of different types of environments and host plants. Some are generalists, so they can be found on multiple islands. Some are really specific to where they are. It's been estimated that there are over 700 of these species. And because they've been studied for several decades already and people have been fascinated with their evolution, we know quite a bit about their diet, their behavior, the environment, and how they're connected genetically. So the advantage of having this kind of model system is that when you have more diversity, you have more opportunities to correlate their lifestyle and habitat with microbiome function. There are other advantages to using Drosophila. So they have a number of conserved cellular and molecular features between mammals and insects. So these images just show a mammalian gut versus an insect gut. Both of them are segmented in similar ways, and based on the different colors, we have different segments playing kind of similar roles. Just as with mammals, different microbes inhabit different parts of the gut and contribute in the same way to the physiology of the host. At the cellular and molecular level, the cells that line the gut play a number of similar functions between mammals and insects. They provide a physical barrier from pathogenic bacteria. In both types of systems, there are cells that secrete mucus that offer this kind of protection and immunological defense. Both organs undergo self-renewal, so you can find populations of active and quiescent stem cells. And both types of guts have enterocytes, which are specialized cells that secrete hormones. And some of these hormones are like leptin or induce the secretion of insulin, and this helps with appetite control and energy balance. So there's a lot in common between mammals and insects, and we can learn about how the mammalian gut functions by doing a number of simple experiments using insects. The advantages of using this simple model system versus humans or mammals is that we have a much simpler microbiome in the fly. So we have usually between 6 to 30 taxa. 6 if it's a lab fly, so the flies that are raised in captivity have simpler microbiomes, whereas the ones we find in the wild have slightly more complex microbiomes. We can also control their lifestyle very easily and do experiments at particular ages, manipulate the diet in really specific ways, as well as the environment and how much activity they get. And this is in contrast to humans or mammals, where you would have to control for a number of environmental as well as lifestyle differences. And if we are interested in fungi and the interactions between bacteria and fungi, yeast are a lot more common in fly guts and play a more intricate role with the fly biology compared to humans, where we don't actually know that much about fungi. And what we do know is mostly with respect to their pathogenic influence. So using all these advantages of the fly, and specifically Hawaiian Drosophila, the main research aims in my lab are to address how bacterial-fungal interactions play a role in adaptation and evolution, and to look and identify mechanisms of bacterial-fungal interactions and their contribution to host physiology. So we use two approaches to do this. One is that we survey the microbiome of naturally diverse populations, such as Hawaiian Drosophila. So we collect the flies from the wild, we characterize their microbes, and then we try and correlate the community with their evolutionary history and their lifestyle. And the second aspect of work in my lab is to perform this kind of experimental manipulation of lab-raised flies. So by bringing them back into the lab, then we can manipulate their microbiome and look at traits, life history traits that are important for their evolution and survival. So this is how we collect wild flies. My postdoc in the lab, Matt Medeiros, has diligently gone out to 6 of the major Hawaiian islands and collected flies from various sites throughout in the forest. So these are just examples of some of the ones that have been caught on Kauai, on Maui, on Oahu, and certainly in the HETF at Laupāhoehoe on the island of Hawaii. So how we do this is look for their host plants. So these are examples of some of the host plants, including Claramontia, Olapa, and Wickstromia. Then we put up these sponges, and the sponges we smear with banana and fermented mushroom juice. And then you can see here from the temperature monitor that usually we want a good temperature around 19 to 22 degrees. So it's a little bit cooler where they are, and it's a little bit more elevated and tends to be quite humid. So then once we set up the baits, we just sit and wait for them to come. And then you can see a couple here on the sponge. And once they get there, if we're fast enough, then we can catch them by putting a vial over them. So we've managed to do this for a number of different islands and sites, 18 sites, 6 islands. And so far we have at least 34 different species. So then the next step is to bring the samples back into the lab and look at their microbiome. So to do this, we first extract the DNA. So we take the fly, we mash it up, and then we put the buffer in there and extract the DNA from the rest. So the DNA that we're collecting consists of both fly DNA and bacterial and fungal DNA. In order to just look at the bacterial or fungal DNA, we do an amplification step using primers that are specific to bacteria or fungal genes. So one specific gene for each of these kingdoms. And this is called amplicon sequencing, and it's PCR based. And basically we're just looking, each bacteria or fungi has a specific barcode, and we are reading this barcode from the collection of microbes within a single fly. And then we put this through a sequencer, and the sequencer reads out this short sequence. It's about 400 base pairs. And then we match up this sequence against the database of known microbes where other people have deposited their own barcodes. And then we match up the sequence and can get at least a genus level identification. And then based on how frequently certain barcodes come up in this amplification process, then we can draw a pie chart or make a graph of the relative abundance of one type of microbe versus another. So we do this for bacteria, and then we do a separate kind of analysis for fungi and come up with these relative abundance charts. And this is our microbiome profile. So what happens when we do this with the Hawaiian flies? So based on an initial snapshot, we divided the types of flies we were collecting into 3 broad groups. One are Hawaiian Drosophila picture wings, another are Hawaiian Drosophila non-picture wings. So both of these are endemic species, but one is from this picture wing subgroup and the other is not. And then non-Hawaiian Drosophila, and these would be invasives like Drosophila suzukii. And these bar charts here show the relative profiles of bacteria and fungi, and the different colors correspond to different genera of each of these microbes that we find. And you can see from the different colors between the Hawaiian Drosophila, the non-Hawaiian Drosophila, and the lab flies, there's a lot more diversity, many more colors in the Hawaiian flies and in the wild caught flies compared to lab flies. And this has been known for a while. And that figure is because we give them just a really simple diet in the lab. And a similar trend goes for fungus. And when we do a number of diversity measures indices to look at the difference in diversity between these broad groups of flies, what we find is that overall bacterial diversity is greater between each of these different groups. So there are more diverse groups as well as different proportions of these groups when we compare between these three major classifications, and that goes for bacteria. For the fungi, there's a little less diversity in terms of how much it changes from group to group. So it seems that bacteria, the bacterial profiles change a lot more, whereas the fungal ones are a bit more stable if you're comparing group to group. When we do this sort of comparison and we classify these groups of flies by island where we caught them. So this is again an initial snapshot based on our first set of data. So if we compare the profiles of flies caught on Hawaii, the island of Hawaii versus Kauai versus Maui, we can see three separate groupings. And again, so just below here are diversity indices. And it shows that for the bacterial barcodes, we just get a lot of diversity, both in abundance and number of taxa and number of different taxa from island to island. Whereas if we look at the fungal barcode, also called ITS, we see a lot less of this diversity. So again, it repeats the point that the fungi diversity is not that different, or is not that subject to change based on the environment. If we do this sort of analysis by classifying the groups by what we know about their diet, there are no significant differences in their relative abundance or in terms of who's there with respect to both bacteria and fungi. So what this tells us overall is this diet, in this case, doesn't matter. They all look similarly or equally diverse from each other. So if we can conclude one thing so far, it's that the environment plays a big role in shaping how the microbiome assembles, and there are big differences from island to island, and these differences are mostly in the realm of the bacteria rather than the fungi. So this is just an initial snapshot, and we're going to add in a lot more data that we acquired in the past year. So another way to look at this kind of complexity of data is to make predictions about which microbes are important for the host and which microbes tend to show up more often than others. And we do this using a form of bioinformatic modeling. So this is done in collaboration with Laura Tipton and her student Chenua Falatoi at Chaminade University of Hawaii. And what they do is that they take all these data we give them from the profiles, and they make predictions about which microbes tend to show up when another microbe shows up. And specifically, we're looking for which bacteria and which fungi co-occur. So just because something co-occurs doesn't necessarily mean that they are interacting with each other, that they are helping each other out or being symbiotic with each other. But it does give us a way to make a prediction about which microbes are likely to interact. So on the left here, you see such a co-occurrence network. Each of the individual shapes called nodes represents different families of bacteria or fungi, and the lines or edges that connect the nodes are the possibility of their interaction. So some of these shapes are really well connected and have multiple connections and are pretty dense, and the connections are short, meaning a greater likelihood, and others are more disparate or have no connections at all. So then we identify the taxa that contribute to the centrality, to contribute the most to the hubs of this network. And then based on this distance from each other, or short lack of distance from other nodes, we can predict which could be putative keystone species. So this is the idea that particular taxa within this community of microbes contribute the most. And if you don't have these species, you may not have a balanced microbiome, or you may have a microbiome that is dysbiotic and not good for the animal. And again, these are all predictions, but at least it targets a few species from hundreds, and allows us to start going after these species, trying to isolate them in the lab, and then look at them and see whether they really do contribute to the host physiology. So to summarize this first part of the talk, what we're seeing from our collections in the field so far is that in Hawaiian Drosophila, the bacterial profiles exhibit greater variation, and we find differences between islands and between their evolutionary lineage. Fungal profiles also differ, but they are less taxonomically diverse than the bacterial profiles. And predictive network modeling points to putative ketone species of bacteria and yeast that could be critical for the assembly of the microbiome in the host. Currently, we are adding more sites, more samples, and both endemic and non-native species to our analysis. And we're trying to isolate some of these microbes from the gut for both in vitro and in vivo culture experiments. So that's the first part. So with respect to the second aspect of my research, that's what I'm going to talk about now. So this is experimental manipulation of flies that we bring into the lab and do a number of different manipulations of their microbiome under controlled conditions. So to do this manipulation, our strategy is to feed antimicrobials to one species of Hawaiian flies, and these are Drosophila polypes. And this is just a contrast with melanogaster, which is the standard lab model. These are not to scale. Polypes is at least 2 to 3 times bigger than melanogaster. These flies, we feed them antibiotics, antifungals, or a combination of antibiotics and antifungals. You can see this is how we raise the Hawaiian Drosophila. They're in these large pickling jars, and they like a particular type of sand where the larvae will burrow into the sand before pupating on the sides of the bottle. Whereas with regular lab flies, inbred lab flies that we grow, we just put them into a vial of food and they grow, and we don't have to separate them. We don't have to give them special food at different ages. So they're a lot easier to maintain, but also in many ways less interesting, I think. So once we feed them these drugs, the Hawaiian flies, then we measure features that are important for their life history and evolution. And this includes lifespan, fertility, stress resistance, and fat levels. I'll just give you a quick overview of how we do these assays. So with the lifespan assay, we put flies, individual or by pairs, into food containing the various drugs, and then we flip them every 7 days or every 7 days into fresh vials. And we do this and monitor how many flies die in this time. So they usually live for about 2 or 3 months. So we have to do this pretty continuously. About fecundity and fertility, we do this by allowing flies to mate for 48 hours, and then we flip them into fresh vials, just the females alone, every 4 days, and then we count the number of eggs that are laid and the number of adults that emerge. And then with respect to stress resistance, we measure this by putting flies into what's called a desiccation chamber. So it contains a foam plug, it contains desiccant, and it's sealed at the top. The flies don't have contact with the desiccant, but they're put in this really unhumid chamber, which is like 0% humidity. And then this is just a picture of what it looks like with all the vials set up with individual flies in the vials. and then we monitor survival every 30 or 60 minutes. So what are the results of these assays? So when we feed the flies antibacterial drugs and measure lifespan, this is the lifespan graph showing how many are alive. In this case, it goes up to 25 days. You've taken it to 60 or 90 days. And on the y-axis is the probability of survival. So if all flies survive, it's 100%. And you can see by 25 days, and I can tell you by the end, there's very little difference between the control and the antibiotics fed flies. So antibiotics don't seem to have an effect on their lifespan. If we look at other features like mating success, what we see is that when we feed females antibiotics, the proportion that lay eggs is much lower compared to the controls in the black here on the left. When both females and males are fed antibiotics, we see very, very few eggs that are laid. So it seems to be that the antibiotics have pretty strong effect on their mating success and possibly their fecundity. When we look at stress resistance, so this is in those desiccation vials, you can see again, like over the course of 20 and 25 hours, the antibiotics fed flies die a lot sooner than they do relative to the control flies. And the way we read this chart is to look at the 50% death rate and to measure how much time it takes for one condition to reach 50% versus the other. And you can see that the antibiotics fed flies, 50% of them die a lot sooner than in the control population. And finally, we started measuring some of the fat levels and other kinds of metabolites. And we see that total fatty acids in the antibiotics fed flies is actually higher than in the control. So this indicates that by eliminating the bacteria, we somehow imbalance the metabolism of the fly. And this may be related to the fact that these flies lay fewer eggs, are less likely to mate, and less resistant to desiccation. So bacteria seems to have a large effect. What about fungi? So when we feed them fungi, again, we see that we have a similar lifespan. They're both dying around the same time and around the same rate. With respect to mating success, again, when both females and males are fed antifungals, the number of eggs that are laid are far fewer than compared to the controls here on the left. When we look at stress resistance, it's even more striking than it is with eliminating bacteria, in that the antibiotics fed flies are dying a lot sooner. Antifungal fed flies are dying a lot sooner than the controls. And fatty acids show the same pattern, where the levels of fatty acids increase when they're fed, when we eliminate a lot of the fungi. So again, this shows that some sort of imbalance of the microbiome is critical for the physiology of these flies and maintaining some kind of homeostasis that allows them to resist stress and also gives them the necessary nutrients for egg laying and for mating. So if you think about this with respect to evolution and how important it is for a particular species to be close to the microbes that benefit it most, you could start to make a hypothesis about how the microbiome and the influence of it and could drive evolution in a certain way, could drive adaptation of the flies to a particular diet or to hanging out around a certain spot in the forest and to become adapted to it so that it can acquire the benefits of these microbes, because without it, it seems like it would have some pretty deleterious effects. And this is also an important consideration with respect to conservation. So a few of these species, not the ones we work on, are endangered. And even though we might be able to rear them in the lab, if we were to transplant them back into the forest, it indicates that we might either need to boost their nutrition with some kind of probiotic or make sure that they have enough of the right microbes around to support their survival. So to summarize this part, microbiome dysbiosis leads to both lower stress resistance, lower mating success, and higher fat levels. And this has implications for conservation as well as their adaptation. So the importance of microbes in insect physiology and their survival, has been known for a few decades. This was first observed in cactus-adapted Drosophila. So these are flies that live on really specific species of cactus found in the Sonoran Desert. And a lot of work from Jim Holdeman and Teresa Markal has shown that yeast from these specific cacti actually help with the physiology of the flies by increasing their development, their nutrition, and the number of them that emerge from pupal form. In other cases, also with ants and beetles, these insects rely on these bacterial-fungal interactions for both their diet and as a kind of immune defense. So these creatures will actively farm microbes. They'll farm microbes that are beneficial to them because these microbes will provide nutrition for them, and they will, in the case of some of these leafcutter ants, suppress the growth of parasitic fungi. So this image on the right here from Cameron Curry's lab shows one of these leafcutter ants. It actually has a specialized organ that uses this white patch here to cultivate these fungi. So that's pretty remarkable to have this sort of very early agrarian behavior in insects. So finally, with respect to our microbiome studies, here I've showed you experiments where we manipulate the microbiome by eliminating certain classes using drugs. What we're really hoping to do is to make germ-free Hawaiian Drosophila. And this is difficult for a number of reasons, but this is just very preliminary data showing the difference between a germ-free larvae and a normal larvae. And you can see that there's just a remarkable difference in the size between these larvae and ones that have not been treated so that they keep their microbiome intact. So then this will be a useful experimental tool for us in moving forward and try to put back individual keystone species and see their contribution to the microbiome assembly and the physiology of the host. So now with the time I have left, I'm just going to shift to a slightly different topic, but one that also uses Hawaiian Drosophila and takes advantage of that diversity. So actually, my background is in looking at chemical communication and pheromones. And this is one of the reasons I got interested in Hawaiian Drosophila, is because I wanted to look at the diversity of pheromones amongst different Hawaiian species. Insects are the masters of chemical communication, and they use very different types of pheromones from species to species. These are just some examples of them. These are small molecules that are mostly lipids. So they're carbons, hydrogens, and oxygens. And these are examples of ones of sex pheromones that have been isolated from various pests and other creatures that are just interesting for us. These lipid pheromones control a lot of their important social behaviors, including courtship and aggression. So with flies, if you have the right combination of pheromones, which are aphrodisiacs found on females, then the male will start to mate the female. But if you have male-specific pheromones, this causes males to attack each other. This causes aggression. So this simple shift in the types of pheromones that are around can radically change the decision to either mate or to fight. Shown here, labeled with green fluorescent protein, are pheromone glands that line the abdomen of the flies, and these glands produce pheromones as well as the waxy coating that protects the flies from desiccation and from microorganisms in the environment. So I was curious about what are the drivers of diversity, chemical diversity. And just as with the microbiome, diet can have a large effect because many of the precursors for these pheromones are derived from diet. Abiotic features like temperature and humidity can impact the types of chemicals that appear, just as it does the microbiome, the types of microbes that are found. And then the microbiome can also have an effect on on how these pheromones are produced. And actually in cockroach, it's been shown that if you eliminate the microbiome from cockroaches, you can change the levels of the aggregation pheromone. So it seems that they use some kind of precursor to help make this pheromone in cockroaches. So one way we study pheromones in the lab is using mass spectrometry. This is a system for measuring the mass of chemicals. So because these pheromones are lipids, they don't have a genetic template. We can't sequence them like we do microbiome fingerprints. And so to analyze pheromones, we use mass spectrometry, which is basically a way to weigh them, to measure their weight. And based on the mass accuracy of this, we can predict what is the elemental composition. So we can dissect out individual glands, individual pheromone glands, as shown in this image here. One male-specific gland in Drosophila is the ejaculatory bulb, and we can place it directly in the sampling grid and put this directly into the mass spec and look for the chemical signature that comes from the pheromone gland of these flies. So I wanted to apply this method to look at pheromone diversity across Hawaiian Drosophila, as well as other types, as well as other groups. And on the right here are mass spectra. So each of these different peaks represents a different chemical structure that is found in the pheromone gland of male flies. And I've organized them according to whether they come from the Hawaiian picture wings, another big group, the Repleta group, or the melanogaster subgroup. And you can just see from the patterns of peaks here that they all look very different from each other, indicating that there is, similarly to the microbiome, great diversity in the chemical profiles of the pheromone glands. We take a closer look. So Drosophila sylvestris, one of the Hawaiian picture wing flies, has this really incredibly rich and complex pheromone gland profile compared to lab-raised melanogaster, which is quite simple. Similarly, if we compare sister species, they don't necessarily look similar to each other in terms of complexity or the types of chemicals that are there. So one would not expect that closely related species would have similar kinds of pheromones, which is interesting because it implies that there are other mechanisms other than genetic ones that impact what kind of pheromones are produced. To take a look at this all together, we can classify their pheromone profiles according to different types of chemicals that are found, how many oxygens there are, and whether they belong to sterile groups or are likely to be fatty acids. And basically, there's no pattern that I can see so far. It just seems that different groups are found in different subgroups of flies. Yeah, and it's just going to take a bit more profiling, I think, to be able to figure out if there's any pattern in the diversification of these pheromone glands. One other bonus reason why looking at Hawaiian Drosophila is fun for this question is that Hawaiian Drosophila, unlike melanogaster and other common flies, is that they appear to have a secret pheromone gland. And this was described in this paper from the 1960s. And that's been the only paper on it so far. So if we take, in this case, it's Drosophila grimshawi, you can actually get it to extrude this gland from its butt. So you don't see it here, but Matt, my postdoc, is actually squeezing around the abdomen, and this is forcing out this gland from the butt of Drosophila grimshawi. Below here are scanning electron micrographs showing a close-up view of the fly butt. And you can see that there are a lot of hairs here. And this is the perspective of what we're looking at here from the Hawaiian fly. This work was done in collaboration with Tina Carvalho and Sophie Nguyen, who's a high school student. So there's a lot of detail here. Each of these hairs is probably important for either chemoreception or helping to spread the pheromone around. So if you observe these flies and you put them in a dish, you see them dragging, the males will drag their abdomen and leave streaks on the bottom of this petri dish. And what we think is that it's either an aphrodisiac or some kind of aggregation pheromone for other flies. we took a closer look at this gland. So we made slices into the fly. So we fixed the fly and then made these ultra thin slices along this plane here. And what you're looking at here would be the head, and here would be the tail end. And you're looking from the top down onto the back of the fly. And this here on the right is a close-up view of the butt end, and the features that are labeled with G are what we think are the pheromone glands, the two lobes of this thing that's being extruded. And if we look a little closer, so this is with TEM, then you can see that there's this duct space here where these pheromones, which appear here as these lipid droplets, are likely transported or squeezed into this duct, and these are then emptied out into the gland, which is dragged along the floor. Some other features you can see in here are tracheals, and it's not really clear here, but if you zoom in quite a bit, you can see organelles like rough ER and mitochondria and Golgi, indicating that it is a metabolically active gland. So this is kind of a side fun project, and a number of the Hawaiian species have this gland, a number of them don't, and trying to see what is the difference in the chemical composition of these lipid droplets in the gland, and to see what is its actual behavioral role, and why is it that the Hawaiian flies have this and other kinds of Drosophila do not. Yeah, so with that, I will just conclude with an acknowledgement slide, thanking everyone who has contributed to the work I talked about. So Matt Medeiros is a postdoc in the lab who did a lot of the collections and the microbiome manipulation. Sandra Tafit as a postdoc who did the microbiome analysis, and Matt is stationed with Don Price at UNLV. Don, some of you will know, is also uses the HETF quite a bit to collect flies, and he raises them in his lab at UNLV. A lot of the genomics data was processed by MGAL at UH Manoa. So this is a facility for the processing of microbiome libraries. Microscopy was performed by Tina Carvalho and Sophie Nguyen in the Microscopy Center at UH Manoa, and my collaborator at Shanghai University, Laura Tipton, and Nona Filatoy contributed to the bioinformatics analysis. Finally, I would of course like to thank HETF and DLNR and people at DLNR, Carl Magnaca and Steve Montgomery in particular, for pointing us in the right way to find these flies, to raise them, and teaching us so much about their natural ecology. And yeah, that's it. With that, I'll take any questions.
Thank you so much, Joanne. I don't see any questions in the chat right now, but if anybody has any questions, feel free to put on your cameras or put on your audio and ask away. If there aren't any questions, thank you again so much. That was awesome. Oh, sorry, there's one question in the chat. I spoke too soon. Dan in the chat says a non-academic question. What are some good sources of microbiomes for people?
I mean, you mean beneficial microbiomes, I'm guessing. This is a non-academic answer. I guess it would be yogurt. It would be vegetables that are fibrous. If you eat certain kinds of vegetables that have good fiber and have good precursors, then it will encourage the growth of some of these microbe families that we think are more beneficial for our health than other types. You can take probiotics, you can take these designer pills, but the doctors, what I know of, say that you have to take them often. You have to replenish it very quickly. And they don't actually think that there's a benefit to it. If you have some sort of deficiency, then it can help. But if you're pretty much healthy, have a well-balanced diet, it's not going to make a noticeable difference. That said, it appears that fecal transplants from people who are immunocompromised for some reason, those seem to have great benefits. But that's a pretty extreme case when you're actually suffering from a disorder, and I think maybe what Dan is asking about is sort of just for day-to-day sorts of dietary choices. In which case, yeah, I have heard that you basically just don't buy into the probiotics. Yeah.
Awesome, thank you. And Anna in the chat just says thank you for the very informative presentation. Awesome. Any more last questions before we sign off for the day?
Thank you, Joanne. Thank you so much for your presentation. It was wonderful. And thanks to everybody. Sorry, go ahead.
Oh yeah, yeah. So please follow up. Just shoot me an email if you have any follow-up questions. And if you are on UH campus, let me know. I can show you the flies.
Nice, thank you.
Awesome.
Thanks,
So I
everyone.
guess we can wrap up. Thank you all for coming. Be on the lookout for the follow-up email that I will be sending. And yeah, looking forward to seeing you at the next one. Thank you guys. Have a good rest of your day.
Bye, everybody.
Bye.